Hi everyone, my name is Zach and I'm currently in the final year of my PhD in bioengineering where I'm studying orthopedic biomechanics at the University of Pennsylvania. I'm here in the McKay Orthopedic Research Labs, and to celebrate STEM Week and Career Day, I'm going to take you through what I do day to day as a scientist. I'll give you a brief tour, show off some of the machines and instruments that we use, and just let you guys know a little bit more about the details of what it's like to be a bioengineer. All right, here we are outside of our animal facility where we house and breed our animals. I can't take you guys inside because we do respect the privacy of our animals and we do try to give them the best lives possible. These animals are very, very important to us because they allow us to do the work that we need to do that humans won't volunteer for. So basically, humans not going to volunteer to undergo any sort of genetic engineering or take on a new, very experimental treatment. So these animals provide the tissue that we need to do cutting-edge experiments that will eventually result in all of us living healthier and better lives. In the McKay Labs, we do study a lot of orthopedic biomechanics, and what that means is that we take basic principles of engineering and we apply that to different parts of the body. We do this to figure out if a tissue is healthy, if it's healing well, or even if it's stronger as a result of any sort of genetic engineering that we may do, or as a result of any sort of new experimental treatments that we've created here in the labs. Now I'm taking you guys into the actual lab itself. This is where we will do all of our experiments. And first we'll stop off and I'll show off my bench and all of our benches in lab. And first thing you'll notice is that many scientists are actually pretty unorganized in some of their personal spaces. We can see it's basically a mess, but there is a method to the madness. So for me, what I'll use my bench for is this is where I'll basically set up and prepare for various experiments that I'll be doing. I might do a little bit of dissection or sample prep here on my dissection pad. And then after some preparation, I'll take these samples over to other parts of the lab where we have more specialized machine and equipment, some that's been purchased and some that we've actually made in the lab ourselves. And that's where we'll actually collect the data that we need to figure out whether or not our experiments worked. So here's my favorite microscope in lab, and for me this is one of the most important tools that I have. So I actually do experiments on baby mice. These mice are approximately 7 to 17 days old, so their tissues are too small to work with if we're just using our eyes and our hands. So this microscope and some of these small tools that you see here will help me manipulate the tissue in ways that I need to further prepare the samples so I can get the exact data and get them in the exact form that I need to perform my experiments. Here's another station that you'll see me using a lot. So here we have a couple of devices that we've actually built ourselves in lab. And these are basically things that we've created that we've needed, but that nobody sold a solution for. So since this is a lab mostly full of engineers, we have the tools and luckily the ability to be able to design these types of instruments. So as I had mentioned, I do study very, very small mice, and this is actually, to take you through what this instrument does real quick, this allows us to use a laser. You'll see it appear in just a second. Maybe, there it is. Alright, so this allows us to use this laser, and what we do is we'll place a piece of tissue, we'll use this as an example piece, and we'll put it on the platform here, and we can run the laser back and forth, and that laser will be tracked by our computer here, and that'll tell us exactly how big that piece of tissue is. And it's really important for us to be precise with these types of measurements because the basic dimensions of the tissue will allow us to perform or to apply our engineering and mathematical principles to figure out what the tissue is doing after any sort of experiment. Next, we're visiting our machine shop, and this is actually a really cool and unique place that a lot of labs don't have. So these are the tools that we use as engineers to be able to create all sorts of devices that we need that we can't buy, and it basically limits you to whatever your imagination and whatever your skill set in the machine shop as to what you can create and what kind of experiments can be done. Alright, and finally, this is my favorite room in the entire laboratory. We call these devices instrons, and what these instrons actually allow us to do is we can place tissue inside the instron, and we can either pull the tissue apart or we can push down on the tissue, and that will inform us as to the actual mechanics of the tissue itself. So this is my setup, and this is where you'll find me a lot of times in lab. You can see that we have a tank here, and the tank, we fill it up with what's basically a salt water. We use the salt water because the salt, it mimics what the salt concentration levels that are seen in your body. We also have, this is actually a sous vide back here, and we'll put the sous vide inside of our saltwater tank. We'll turn the sous vide up to the exact temperature that your bodies are regulated at, and then we'll perform our experiments within physiologically relevant conditions. And this gets deep into the controls that you need for your experiments. You basically don't want to test something in a way that's not going to be realistic, or in a way that can't be very consistent between samples. So these tools are very important for us to make sure that the data that we're getting is consistent and can actually be useful for us, so that when we're doing our genetic engineering or whenever we're applying our treatments, we can make sure that we can depend on the tissues reacting in a predictable way within the variables that can be controlled by us as scientists. And then finally, for the instron itself, so what we have here, you'll see this is the load cell, and the load cell is actually what produces the force in the instron. And so basically what I'll do is I'll take the tissues from my baby mice that I've dissected and carefully prepped, and then I will put them inside of the instron, and we'll use various grips to hold on to the tissue itself. And then for me, I'm studying tendons, and tendons always work in tension, which means they're always pulling by transferring the forces from your muscle to the bone. They're absolutely vital for movement. They're easily injured and they don't heal well, so that's why I find it exciting to study these tissues, because everyone uses them and almost everyone hurts them at some point in their life. Alright, now we're headed into the office space, and it's not going to be the most exciting room, but I do consider it among the most important tools that we use in the lab. That's our computer. So I've taken you through the process of where we prepare our samples and where we test the samples, and those tests give us a lot of data that we have to work with and figure out how to take these numbers that we have and how to turn that into a story that's useful for people to understand what's going on. So it does produce a lot of data. We have many terabytes of data. I think we're up to 25 terabytes of data across all of our lab experiments, which is a ton of data. So the computers ultimately help to be our tools to process all that data, apply our math, apply our engineering principles to it, and figure out really if our tissue, if our treatment, if our manipulations have been a success, so that we can better inform ourselves as scientists, and eventually many decades down the road we get from this cutting edge scientific research into some sort of treatment that will help human beings live better lives, have less pain, and live for longer. Alright, so that's the end of our lab tour. I hope everyone got to see some cool new things in the lab and got an idea of what kind of environment a scientist does work in, and learned a few new exciting things about science and the type of work that we do here in the McKay Laboratory at Penn. So I wanted to end by talking about my time training as a scientist, kind of what it takes and what it means to me. So this is my 10th year training to be a scientist, and that's 10 years since I graduated high school. So it took me 4 years to study and complete my undergraduate degree in bioengineering, and now I'm ending the end of my 6th year in my PhD. The PhD is the final degree that's needed to actually become a fully independent scientist. It provides a really strong mentorship where you study underneath a different scientist who's started their career, or maybe even towards the end of their career. They teach you how to properly do your initial research, how to formulate your questions, how to conduct your experiments, and then also on how to communicate what you found in your research to both an academic and even an audience that's not trained in science. So since it does take 10 years to complete a PhD from the start of your undergraduate degree in college all the way through the end, there are a lot of challenges that come along with that. It's definitely not easy, but I do find it very rewarding. I think it's rewarding because as a scientist, you are on the cutting edge of human knowledge. So what that means is that you are going to work every day and you're coming in and conducting your experiments, and over the course of decades, you're basically producing new knowledge for humanity, for civilization that didn't exist before you did that work. And I think it's really cool to look back on what science has done for us as humans, where that's taken us for the type of lifestyle that we've lived over the last really couple hundred years since science has been a thing that humans have really valued and latched onto. I think that to be a small part of that is a really, really cool thing. It is challenging, but at the end of the day, I think that it is worth going through that challenge because I think that there's nothing more exciting than being able to create a new piece of knowledge that makes the world a better place. So I just want to thank you guys for coming along with me on the tour and giving me your time and your attention. And I think that no matter what you're studying, even if it's not science, I think it's really important that everyone does their best and pursues something that they can make an impact on, and everyone helps contribute to make this world a better place overall. Thanks, guys. See ya.